So very good afternoon, all of you. Again, a very warm welcome to all the participants and then the speaker to the second day of International eLecture Workshop on Innovation in Biotechnology. So due to the response from you all, we got that feedback last day. It has contained a lot of improvised information and then suggestions and then appreciation in the comments. Thank you so much for your support. So we're gonna initiate with today's deliberations with Professor Alok Shaw. He's the Director, Sustainable Food Processing Center, Food Future Foods, and also from the University of Nottingham, Malaysia. So we're gonna deliver talk on recent development of microalgal biorefinery. So to talk introduce about him, it's very tough to compile his profile into a smaller version because he have achieved a lot in his career. Some of the few are highlighted here. Actually, he's a Tanstri Emeritus Professor, International Special Award for his innovation and intervention in palm oil recent last year. And he is one of the ranked scientists and top 2 scientists in Stanford list. It's published in the recently in October. And also he got National Young Scientist Award in the year 2019 and Vice Chancellor Award and Global Top Peer Reviewer Awards in the various domains in engineering, biology, biochemistry, and chemical engineering as well. And got an Da Silva Award from the Society of Biotechnology Japan and Asian India Research Training Fellowships, GSPS Fellow Awardee, and also the Top 100 Asian Scientist Award, and also he is a Rising Scientist Award. So with that, he's specialized in the field of bioprocessing from upstream to downstream field and separation purification technology and algae biorefinery engineering. So these are his recently running special issues from various journals. You can see the renewable and sustainable energy reviews, environmental pollution, hydrogen energy, biochemical engineering journal, energy research. So it's you can see that impact factor of all these journals is 14.8 and then and chemosphere. So many other journals also he associated with his publications. So better to explain him, I gave his link. You can go through that. Me take 2 days of reaching all his credentials in terms of publications and then his award he received. And very much thank Dr. Shaw for his consent to give a talk and this through online to address the young minds through our society. So I'm very much thank and then welcome Dr. Alok Shaw to share his experiences out of research. So over to you.
Yeah, thank you so much, Chairperson, for your kind instructions. Right, please allow me to share my screen now. All right, so you be able to see my screen?
Yes, we can see.
All right, good. Okay, thanks again for inviting me to give a sharing section today. So my name is Alok Shaw. Can call me Alok. I am from Nottingham, Malaysia. Right, so as usual, I would like to give a short introduction about my university. So as you know, my university actually we have 3 campuses. The first campus is located in UK, which around 34,000 students. So for information, UK campus is a government university. Right, our second campus located is in China. People's Republic of China is around 7,000 students. So it's a private university. Yeah, so that's why and together with University of Nottingham, Malaysia, which are around 5,000 students. For information, University of Nottingham, Malaysia, this year is our 22nd anniversary. So we have been in Malaysia for 22 years already. So a little bit background about myself. Right, so my currently I'm the technology advisor for high value, high tech agriculture Malaysia under the Ministry of Malaysia. So whenever there is a research, so I'm the advisor to give an advisor to the government. So I also rank #1 in the Microgame technologies. Currently, I'm the president of International Bioprocess Society in Malaysia. Right. So I also director of Food Processing Research Center in Nottingham, Malaysia. Co-director Future Food Malaysia Beacon Excellence. So many of you maybe wonder what is Beacon Excellence. Beacon actually is a research institute in Nottingham, Malaysia. Right, I just got promoted become a full professor last 2 years ago. So there's in the biochemical engineering, there's a lot for me to learn from you all. Right, so I got my PhD in very fast time, I can say in 2 years time, right after my bachelor degree. Then I used 2 years to finish my PhD. Right, so right after my bachelor degree, then I proceed to master. Then for master, I use another 1 year to convert to PhD. So in total, I used 2 years to finish my PhD degree in bioprocess engineering. Right, so right after my PhD, I will have another degree. We call it PGCHE, which is postgraduate higher education degree. So currently I'm a fellow of higher education academic. So that's qualified for me to teach in the university. Right, I also chartered chemical engineer, which is ICHEMI, professional engineer, which is with Malaysian Board of Engineer. That's why we can see my title with a IR. Right, so I also professional technology, which is in Malaysian Board of Technology. So that's why you can see my title with TS. Right, so this is a brief introduction about myself. Okay, so my area of research has mentioned by the chairperson just now. So my area is covered from bioprocess from upstream to downstream. So upstream maybe is cultivation, fermentation, to downstream which is the separation, preparation, extraction technology. So I during my PhD, I work a lot in separation, preparation technology. I developed a method we call liquid bioprocessing flotation system. So in today's sharing, I will focus a lot in this liquid bioprocessing system. I will share the 3 techniques with you in this section. Right. So later on, I will recently I also interested with Microgame Biorefinery. I work a lot of Microgame Biorefinery. So you can see as Microgame, we have bioprocessing. Microgame requires sunlight, carbon dioxide, and water. Undergo bioprocessing, we produce biomass. Biomass can be wet biomass, dry biomass, and the oil biomass. Undergo biorefinery, we produce algae biocrude, which is one of the byproduct. Then from dry biomass, we produce polyunsaturated fatty acid. Then for dry biomass, also produced biodiesel as well as pigment. For the oil biomass, we produce ethanol. So this is the whole structure, whole concept of this microalgae biorefinery. So today's topic I want to share with you all, which is the latest development of microalgae biorefinery. So in my section, I will divide into what is microalgae, then what is biorefinery, how to combine microalgae and biorefinery. Then I will share the latest development, which is the method developed by our team. We call liquid bioprocessing flotation. If any questions, feel free to raise your hand. Then if you anything you want to ask, feel free to unmute yourself, or you can type your questions in this meeting chat. So I will answer accordingly. Right, so let me begin. Right, what is microalgae? Microalgae, actually, everyone know that actually is, you can see is a something like moles, on top of the legs, or inside the leg, or this way. So microalgae can divide into few category. I can say micro or macro. So it's a photosynthetic plant which can grow rapidly, can go very fast. Can go maybe up to can duplicate up to 12 days, 14 days, depends on the strength that you use. Right, can grow with the help of carbon dioxide, sunlight, and nutrient-rich water. So that's why you can see for this microorganism, right, for this microalgae, only require carbon dioxide, which is not require oxygen. So this is very good. Things can reduce carbon dioxide. Only use sunlight, which is free of charge, and on nutrient-rich water can be wastewater as well. So the ranging from unicellular to multicellular. Unicellular can be very small, one cell, or multicellular can be multiple cell. So size can be 0.5 micrometer, which is very small. We call it microalgae. Can be very big, very large, very long. We call macroalgae. Can be 100 meter, which is in layman term we call seaweed. All right. So this is something macro and microalgae. All right. So as I mentioned to you just now, the composition of microalgae divide into 3 major compositions. The first one is carbohydrate. Protein, lipid. Right. So what can produce from carbohydrate is the bioethanol, biobutanol, and bioplastic. While what can produce from protein is animal feed, human nutrient, single cell protein, pharmaceutical, cosmetic, and biofertilizer. While lipid can produce by the cell and nutraceutical. So as I mentioned just now, only require sunlight, which is free of charge, water, which can be any water, wastewater, salt water, fresh water, and inorganic carbon, which is carbon dioxide, and of course need some nutrient as well, nitrogen, phosphorus, and potassium. Right. Other than carbohydrate, protein, and lipid, microalgae also contains some valuable bioactive compounds such as vitamin, mineral, pigment, folate, and sulfuric acid. So this is very important and valuable product can be used as food supplement for human being as well as animal. Right. So some interesting fact to share with you is algae is superior to conventional protein. Why I say so? Because algae can have a lot of protein. Of course, the large variation between the different microalgae species. So example, you can see spirulina around 70%, while others we have only 10%, while chlorella only have 58% of protein. So different type of microalgae have different type of protein. But compared to other crop, right, you can see microalgae contain 10 to 70% of protein. Right. So another interesting fact to share with you, which is land area required for producing 10,000 of protein, 10,000 kg of protein, only require 0.7 to 2.5 hectare of land, compared to other crop which is required, let's say, rapeseed require 32.8 acre of land to produce 100,000 kg of protein. So in other words, microalgae require less land, which is less competition because human being we have getting more and more population. That's why you can see less land is more important now. Okay, so this is some interesting fact. Also to compare with other crop, right, algae protein. So you can see microalgae have more protein, more lipid, and more compared to other crop. Let's say you can see, we have 58,000, while other crop like corn, soybean, sunflower, palm oil also have less yield, right? So land required also less because microalgae only grow in water, and while the cell production is very high compared to other crop. So this is some interesting fact to share with you. All right, so let me share with you a quick video, right, to let you see what is overview of microalgae, how microalgae benefit to human beings.
Sustainable development is defined as a development that allows us to meet the needs of the present without compromising the ability of future generations to meet their own needs. However, the reality is that we are far from that. Never has the impact of mankind on the planet and its ecosystems been so tremendous as during the last 100 years. This circumstance is critically worsened by an exponentially growing world population increasing at a rate never witnessed before, putting food supply certainty at great risk. How can we produce and supply enough food in a sustainable way to a population which is expected to rise to almost 10,000 million by 2050? How can we produce 60% more food than today in just 3 decades when the climate change and eroded soils are greatly affecting agricultural productivities? With this critical scenario, we need to find innovative solutions to help us tackle the problems. In this context, microalgae have real potential to contribute towards the sustainable development. But what do you really know about microalgae? Microalgae are microorganisms that first appeared on Earth around 3,000 millions of years ago and created the atmosphere as we know it today. They are the first organisms that performed oxygenic photosynthesis and are therefore the origin of all known plant-based life. They are the reason why we have oxygen on Earth. And still today, microalgae living in the oceans provide us with more than half of the oxygen we have. Also known as phytoplankton, they are the first link in the aquatic food chain. However, we have not paid much attention to them until now, because we now know that they have great potential to improve our lives in many ways. There are more than 60,000 different species, all of them with different biochemical composition and properties, hence with many different potential applications. Furthermore, they are highly productive, even allowing a daily harvest. They don't compete with conventional agriculture since they don't need fertile lands to be grown. And they are an inexhaustible source of biochemical richness, including proteins, omega-3 fatty acids, carbohydrates, carotenoids, polysaccharides, vitamins, or phytohormones, among many others. Thanks to this composition and their biodiversity, they are a raw material with immense potential for application in many different sectors. Microalgae can be a key part of the solution because they are a source of clean, sustainable, and inexhaustible biofuels. Because they are one of the most efficient CO2 biofixation systems in the world. Because they are a novel food source cataloged as the food of the millennium by the FAO of the United Nations. But more importantly, microalgae-derived products are already playing a key role in considerably increasing productive yields in agriculture, aquaculture, and animal production. For all these reasons, we not only owe microalgae life as we know it, but also the improvement of our lives in the coming years. They are our origin, but they are also our future.
Okay, I believe you have watched this video. All right. Okay, right, let me proceed with the next slide. What is biorefinery? All right, so whenever people talk about biorefinery, what is biorefinery? Actually, there is a two definition well-defined biorefinery. So the first definition defined by International Energy Agency, which is IEA. Second definition by NREL, which is National Renewable Energy Laboratory. Right, according to IEA, which is a biorefinery is a sustainable processing of biomass into a spectrum of marketable products and energy. So this is a definition by IEA. While second definition by NREL, which is a biorefining is a facility that integrate biomass conversion processes and equipment to produce fuel, powder, power, and chemical from biomass. So you can see both definition also involve biomass. Yeah, both is also involve variable products. So example, variable product can be fuel, power, and chemical. So both also almost the same thing. So just a brief definition of microalgae biorefinery. As I mentioned to you just now, but in algae biomass, we have lipid, we have protein, we have pigment, we have carbohydrate. So undergo a pre-treatment to break the cell wall, because in microalgae is a plant cell, right? You have very thick cell wall. When you break the cell wall, all the active compound inside the cell will release, right? So you can undergo the pre-treatment can be sonication, microwave, enzymatic, hydrothermal, pulse electric, and also thermal shock. So for this, then for carbohydrate, we undergo fermentation, we produce hydrogen, methanol, ethanol, lactic acid enzyme. While for lipid, undergo transesterification, produced by the cell. For the undergo for glycerol, you put the fermentation, produce protein lactic acid. While protein undergo a certain treatment, you produce a fertilizer, some example. Well, this is some residual of it. Then from the wastewater, which is an influent, then you can reuse back to grow the microalgae, to cultivate the microalgae. And this is how we call it circular economy, to recycle everything, to have zero waste discharge. Right. So what is the future of biorefinery? So everyone talk about biorefinery. According to first generation of biorefinery, which we focus on food feedstock, which is sugarcane, corn.
Okay, sorry for the interruption. There is some technical issue because he is from he's sharing from Malaysia. So please, just that you're going to join shortly. So Professor Shaw will join shortly because his computer restarting suddenly. Okay, after this talk, we're gonna have inauguration of association called Nano and Biomedical Association. We're gonna some professor gonna give a illustration for that, and after that, I'm gonna share you a feedback link for this day 2. So whoever is available in the Zoom rooms and in the YouTube link, they can give the feedback, and then after that we take a week time to send you the certificate. You cannot expect next today, so please you have to wait. If not, then you can mail us if you are not received, because some of you are not submitted the right mail ID. Maybe some dots is missing, or the extra letters. Because of that, I could not communicate for the sending these links. So to adjusting these things, we have to wait for a week after the submission of the feedback link. Okay, otherwise you can mail me or WhatsApp me in the group. Then I can we'll see that your participation, and then we send you the certificate G course. I hope you understand this technical issues. So yes, he's joining now.
I'm sorry for the technical error just now. I'm back now.
Yes, thank you. We can continue.
Right, sorry again. Okay, let me continue with this. Yeah. Let me share my screen again. All right, I hope you can see my screen again. All right, so when we talk about our first generation biorefinery, we focus a lot. We talk about food feedstock, sugarcane, corn, and kawas. All right, so while it's come to year of 2000, we have second generation biofuel. We talk a lot like another biomass. Example, we have forest, yeah, agriculture, agro industry, municipal, and also some specifics. While coming to third generation of biorefinery, we're now focusing a lot of this algae feedstock. You can see everyone talking about algae because it's just third generation of biorefinery. While come to the closer one, which is fourth generation of biofuel, which is biorefinery, which is talking about genetic modification of raw material. So we modify something that become more powerful. While what is fifth generation biorefinery? I think everyone is discussing about it right now. So what is fifth generation biorefinery? So just to let you have a better understanding of microalgae biorefinery, actually microalgae actually start from microalgae culture cultivation. Then we add in the biorefinery concept, which is a protein, carbohydrate, lipid, and pigment. Then we become a market product, which is high value product commodity, example which is biofuel. So this is the overall concept of microalgae biorefinery. So we combine microalgae together with biorefinery, come to a market already. Right. So I would like to share with you what is artificial intelligence as well, because now it's a hot topic, especially in Malaysia. We talk about Industry 4.0, Industry 5.0, then we incorporate this industry, artificial intelligence. So what is artificial intelligence? Actually, artificial intelligence is a refer to a simulation of a human intelligence in machine that programmed to think like a human and mimic their action. So this is something that is working right now. So in the first industry revolution, we focus a lot is steam engine. Second industry revolution, we focus on electric motorized, which is assembly line, say conveyor belt. Third industry revolution, we use a lot of automatic arm, robot arm, all this or automatic. So fourth industry revolution, we focus on production of system, which is that learn, adapt, and practice, which is using cloud system. Internet of Things. All right, so we can look something into it right now in this research. So we are doing something new biorefinery technique we call liquid biophasing system, which incorporate this microgame biorefinery and same time using IR in this system. We call liquid biophasing system. What is liquid biophasing system, by the way? Liquid biophasing actually is a liquid partition method of two liquid phases. Right, so we successfully recover and separate ionic active compound. Example, my active compound is color, pigment, protein, carbohydrate, lipid, vitamin, mineral, enzyme, nutrient, aroma, heavy metal, nanotube. So whatever that you name it in this system that we can do the separation. So we are successful published a lot of paper with this method. So we can say we are the first person who developed this method. So no matter whatever the limit, we can use this liquid biophasing system to do the separation and purification. Right, so how to prepare this liquid biophasing system? Just very simple, just how you make coffee. You make coffee, I believe you need sugar, creamer, hot water, and also coffee powder. So you make it, place the undergraduate force acidification if you need fast result. So how the mechanism to working on this liquid biophasing system? Actually, there is two main factors affect this liquid biophasing system. The first factor actually is a phase forming component, just like how you make coffee. Coffee has Arabica coffee, Robusta coffee, different type of coffee. The ingredient, also the type of material you use is important. That's why phase forming component is the most important in this liquid biophasing system, because the phase forming component can be as a self-destructing agent as well as separation and purification agent. So how this phase forming can be as a self-destructing agent? So you can see this is the phase forming component. Can be alcohol, can be sugar, can be ionic liquid, can be salt, can be polymer, different type of phase forming component. While you put the phase forming component inside on it, right, you can see you attach on the surface of the cell. Right, you will break the cell wall, then you release all the active compound. This is because self-destructive agent. Second one is the phase forming component can be salt, can be sugar, which you put the second phase forming component, then you will put inside here, then you can see all the second phase forming component, all the active compound will partition to the top phase. So this is how we perform the separation and purification agent. Right, so this is how the liquid biophasing system works. The overall concept is very simple. While second main factor system, which is the concentration of the phase forming component. Why is so important? Because below critical concentration become visible or homogeneous single phase, while above the critical concentration become turbid, which form two phase. So what we want is form turbid and form two phase. So whenever you do the liquid biophasing system, right, you need to construct a phase diagram. So this is the PEG, which is polyethylene glycol dextran. So this above the phase diagram, you will see a two phases. Below the phase diagram, we see a one phase. So this is the timeline length. So you need to form a strict timeline length in order to do the liquid biophasing system. So this overall structure of it. Right, so why liquid biophasing system is so important? Because everyone, when we talk about bioprocess, we want upstream and downstream. So upstream normally focus on fermentation and cell harvesting, while downstream focus in self-destructing, purification, and polishing. Right, just to let you know that total manufacturing cost of this downstream process consists 80% out of the whole bioprocessing line. So you can see this is very important. So you like or not, you need to find a solution to solve it. So the solution that the proposal that we pose is liquid biophasing system, which can minimize the cost as well as to make the system faster. Right. So the benefit using liquid biophasing is high water content up to 85% of water content. Second is low interface tension, easy partitioning, just not like oil and water. Oil and water have high interface tension, but this liquid biophasing system is low interface tension, so easy partitioning from top phase to bottom phase and the bottom phase to top phase. So easy migration, easy moving of the active compound in the interface. So low interface tension is very important. Like rapid processing, I don't think you make a coffee more than 10 minutes, right? Just mixing and settle down. Yeah, maybe less than 10 minutes you can settle it. Right, stabilizing effect, special property of the phase forming component. Yeah, the chemical they use, the phase forming component they use is very stable, do not have any harmful to this active compound. Large scale application, at least 10 liter. In fact, just mixing of water, you can scale up to 1,000 liter. It doesn't matter. right? Simple operation, as I mentioned, mixing and shaking only, right? In fact, we develop a method where we do not require to have shaking and shaking. Minimal energy required, shaking and centrifugation. You can see later in the aqueous flotation system, we developed this system not required to have shaking and centrifugation. Low investment cost, where the chemical that used can be recycled, right? Environment friendly, is no toxic free component in this system. That's why you can use in a lot of pharmaceutical, a lot is this chemical, a lot of this food industry. So we already proven it's high yield, high recovery, high purity. Right. So we have to develop an improved version of liquid biofreezing system, which integrate with this thermosensitive polymer. We name it as a thermal separating biofreezing system. Second one is we integrate with bubble. We call it liquid biofreezing flotation system, which is not required to have a shaking and centrifugation. Third one is integrated with upstream and downstream, which combining upstream and downstream together, which we call it extractive technology in liquid biofreezing system. All this we incorporate this Internet of Things, artificial intelligence in this system. Right. So let me give you some example of how this thermal separating biofreezing system work. Right. A very simple, just like mixing the two liquid phases. You can see this red dot bioproduct plus this thermal separating polymer. Thermal separating polymer can be EOPO, can be different type of thermal separating polymer. You heat it above the critical solution temperature. In that case, you cannot heat too high, because when you heat too high, right, you will denature the active compound and maybe the protein will die. So what you can do is you cannot heat too high, maybe 20 to 40 degrees Celsius, you form two phases. On top of it, you can see our water-rich top phase. Bottom, you can see EOP, alternative oxide, carbon dioxide. Bottom-rich phase, you can recycle. So this is how this thermal separating biofreezing system work. Right, very easy concept. Second one is a liquid biofreezing flotation system. Right, liquid biofreezing flotation actually combining concept of solvent sublimation to go together with liquid biofreezing system. So this is a bubble concept. So this is a gas passing through a cylinder lattice, we form bubble. So we combine this bubble concept with this liquid biofreezing system. We come up with this liquid biofreezing flotation. This gas passing through lattice, we form bubble. So you can see with this concept, right, you can see the hydrophilic with actually attached the active compound at the surface bubble, then bubble will flow to the top. Right, this is how we work, okay? We call liquid biofreezing flotation system. Generally, liquid biofreezing system perform better separation because have performed two layers of separation. The first layer actually is the attachment of the bubble. We perform first separation. Second separation happen in this interface, which is a phase forming component together with bottom phase. Then perform second separation at the same time. So purity and separation more efficient compared to thermal separating biofreezing system. Right. So you can see even it's very simple, but yeah, of course, yeah, we need to have a lot of optimization, a lot of parameters need to do. Just like how you make coffee, you need to have correct concentration of water, correct concentration of sugar, correct amount of water that you put inside. Yeah, this is exactly the same thing. You need to have correct molecular weight of polymer, correct concentration of salt, correct concentration of polymer, correct pH, yeah, volume of the aqua phase, volume of the polymer phase, amount of the crystal, gas flow rate, flow time. The more parameter optimized, the more pure you get. So this is some example how we do the Microgame biorefinery. All right, this is the carbon dioxide. We pass into a cylinder lattice, we form a bubble. Bubble, we use the G4 porosity, which is certain side of bubble, because certain side of bubble have certain separation. Example, G for lipid need a bigger bubble, for protein need a smaller bubble. So different things have different molecular structures. So this is microgame biomass plus this salt solution passing through then all the lipid will attach to the carbon dioxide, will passing through EOPO. EOPO will transfer and attach to heat above the critical solution temperature. On top of the liquid, you can see a lipid. Bottom is a concentrated EOPO, which you can recycle. So this is the overall concept of liquid biofreezing flotation system. Right. So the last technology that I will share with you is called extractive technology in liquid biofreezing system. What is smart extractive technology in liquid biofreezing system? Actually, it's one student can be more than two, but at the same time, you can perform this upstream, at the same time, you can perform the downstream. So you can see this is a phase forming component plus a feed by the microalgae. Then this is a nutrient. We let it do the normal cultivation process. We let it grow as much biomass as possible. Then after the harvesting time, maybe 2 weeks or 3 weeks, you will have enough biomass. You stop agriculture, you heat it above the critical solution temperature again, 30 to 40 degrees Celsius. Then you will see two layers will form. The top phase we call it bioproduct plus the water. Bottom phase consists of concentrated phase forming component, which is EOPO plus the biomass, which you can recycle. So this is the overall concept of smart extractive technology. How we call smart? Because automatic is using biosensor as well as can be monitored using smart mobile phone. Right. So this overall liquid biofreezing system work. Right, so let me share with you a short video how this smart technology in liquid biofilm system. So basically, it's a simultaneous cultivation and biorefining of microalgae. We're using single-step extraction, which is we cultivate microalgae at the same time, then we do the extraction at the same time. So we recover and do the biorefinery. So after the harvesting time, you can see all this biotic compound, right? Yeah, all this biomass will partition the top phase. So after you heat it above the critical solution temperature, right, we perform the two phases. We run the break the cell wall, you can see all this biomass will partition to the bottom phase. So you can see a thin layer of the oil on top of it. Middle is the water and bottom phase is the biomass, which you can recycle the next cycle. Of course, we incorporate this IoT inside, which is the AI inside. We put the sensor in this layer, put the sensor in this layer, and put the sensor in this layer. Then we can monitor the whole system, make the system more digitalized as well as more systematic and more automatically in this system. This is what we call smart extractive technology in this system. Right? You can see oil layer, water, and biomass layer. Right. So how we do IoT agriculture, algae cultivation and byproduct recovery. So this is how we do it in our lab. You can see we do the cultivation as well as extraction in this system. So this is very things that we do. Yeah. So one of my collaborators from Taiwan, actually he developed a microalgae based circular economy. You can see this is a microalgae, right? So microalgae you develop, control using AI and also IoT system. So everything controlled by this system. So you can see this is a crop, right? When we have crop eaten by this pig, so eaten a pig, we have some waste, can use it to generate electricity. By electricity, at the same time, waste water from the pig farm or food industry, you can use to cultivate microalgae cultivation plant. So this way can, for microalgae plant, you produce algae biomass. Algae biomass, you can extract it to functional algae powder. Example, fucosanine, lutein, azathine, EPA, DHA. All these is variable product from microalgae. So this algae can be used and provided and fermentation. Then you can feed to the aqua feed, can feed into the fertilizer. Then you can recycle again the whole system. This is we call microalgae based biorefinery techniques. Okay, so this is all about the success story from my Taiwan collaborator. So the future will be actually, in my opinion, in my humble opinion, the right combination at the right time in the right place. So that's why I very believe that this is a good opportunity for us to meet up. We have a lot of collaboration in the future. So this is some of my potential research that I'm going, right? I'm one of my students working on synthesis new Ionic Liquids. So new Ionic Liquids used to do the separation as well as do the biorefining of microalgae. I also work a lot in this recovery of DHA from microalgae, microalgae lipid from biofuel, extraction of pigment, which is exotin from microalgae, agarose extraction from seaweed, multi-phase extraction in microalgae, microalgae cultivation in wastewater, then continue extraction cultivation of microalgae using liquid biofilm system, pelletizing of food waste compost for fertilization, production of animal feed and supplement from microalgae, as well as the last one is I produce algae biochar. So this is all my research area because I have a big team. Yeah, everyone working in different area. So I like to acknowledge my 14 PhD students as well as 4 ongoing master. Right. So I also have few students already graduated. Example, Dr. Abdur now working in industry. Dr. Chiu working in Xiamen. Dr. Leong working in Taiwan University. So Lizzie working in Singapore. Dr. Radim working in Nottingham. So Dr. Chan working Tan Chong Hong working in Uniten. Shoma working in Africa. Isabel working in China, as well as Wei working in University Kebangsaan, Malaysia. Then Sharon working in Uniten. Dr. Yu working in industry. Yu Kai-Ling also graduated, Dr. Jerry working in CSI. So all is my good collaborator. Even they are my ex-students, now we are still working together. Right, I would like to thank my international research collaboration as well from Taiwan, from New Zealand, from United States, from China, Japan, Vietnam, also from other universities. Hopefully, we have more collaboration from India side as well. So this year I organized a international conference. We call it POSE, which is organized in 6 to 11 June in Langkawi. So in this conference, we have a lot of great special issue example, which is renewable and sustainable energy review impact factor with around 15, environment pollution, hydrogen, International Journal of Hydrogen Energy, then etc. Right. So I believe that publishing a book is still not enough. Paper is not enough. That's why I'm start to publishing a book. Yeah, starting from 2019, I start to publish my first book in bioprocess engineering, focusing downstream process. Then I focus this liquid piping system because I think that this liquid piping system is very powerful. That's why we need to share with others. Yeah, especially industry. Then continue with the prospect of industry 5.0 in biomanufacturing. So this is a new book published last year. This year we ended the book also. So I also actively a lot of journal, example, journal of hazardous material, environmental pollution, chemosphere, environmental research, scientific report, processes by engineer, clean technology, and environmental policy, as well as I'm the editor chief of this current nutrient and food science. Right, this is my last slide. I'm very responsive. You can feel free to email me, then contact me. This is my group website. You can line me, you can reach me, WhatsApp me, and also Facebook me. Right, thank you so much. I pass the floor back to you. Thank you.
Thank you very much, Dr. Shaw, for your excellent presentation. Instead of any technical thing, then immediately you join and then explain well and different aspects. So now we go. Yeah, now we go to this questions. If you have asked a question in the chat box, I can pronounce, then you can able to answer. So the Praveen Kumar, he asked, could you share? Like actually he wants some reference for the smart extraction procedures. So I can share that the public your web page in the group, and they can collect all this publication related stuff and IF research. So the Tajul, Tajul Vaslin has asked about how algae has settled. I mean, calculated which compound that settled the microalgae totally. I mean, flocculation. You're asking how the algae is flocculating, and what are the compound can be used to settle that microalgae. Complete settlement can be done.
All right, yeah. Actually, we are using the EOPO, which is thermal separating polymer in these flocculations. Yeah, we don't use the name of flocculation because flocculation is bubble concept, but we use a nano bubble. That's why we call liquid biphasing flotation system. Yeah. I hope I answered your questions. Right, so I think we have another question for...
Which type of ionic liquids?
Sorry?
Type of ionic liquids.
Yes, we are using different ionic liquid for information. Yeah, we are working together with one of the Portugal group. Yeah, they are producing my ion liquid. That's why we are testing our ion liquid in this Microgame Biorefinery. We tested many types. Yeah, not only one type. Yeah. Yeah.
Any other question from
hydrophilic? Yes. Yeah, both. Yeah, I can say.
So, how about feasibility of criteria of bioenergy production from marine microalgae and commercialization?
How about the feasibility criteria of microalgae production from marine microalgae and commercialization? Okay, so that is actually, I can say feasibility criteria is depend on the strength. Yeah, one thing is the strength, the tough of the strength is very important. The selection of strength is important because not all the strength can be commercialized. Not all the strength can tough enough to survive to produce enough of bioenergy. So I believe one of criteria, physics criteria, is strength selections. Yeah.
So one from Dr. Rajesh from PNU, he asked Taras chytrids can be lab cultured for the proof of production. Because in the algae, it's useful for production, or can we go ahead with gene engineering system? So which one is feasible for extracting proof from this algae?
Yes, I believe that is possible. Possible. But I have tried it. Yeah, if you have any opportunity to work together in this area, I'm very welcome. Yeah.
So have you ever tried for reducing the cost of separation process?
Yeah, actually, for information, the process that I proposed with liquid biphasing system, right? Actually, I'm trying to reduce the overall totaling production cost, because in traditional separation process need membrane separation, need centrifugation separation, needs chromatography separation. But what I'm proposing using liquid biofilm system plus incorporate this IoT smart system so that you reduce the cost very much. Yeah, I can say. Yeah, this is my humble opinion. Thank you.
Yeah. So any other question for the participant? You can raise your hands. I can unmute you. You can ask him directly also.
Yes, feel free to unmute yourself, or you can type in the meeting chat. Then I can answer your question.
Yeah.
Again, sorry for the technical error just now. I don't know, my computer suddenly restart and this come to some technical error. I hope you don't mind.
Yes. So in the YouTube live streaming also, many have appreciated your presentation. No questions were asked. So you can also see there. Fine time. Okay. So I have one question that how the extraction only of using zero wastage can refer as a circular economy, or is there anything that we have considered for mentioning that the microalgal based things are circular economy?
All right, yeah. Actually, it's very good question. Yeah, just to let you know that because some you can see some of my slide, right? Recycling system, yeah, zero waste. Actually, it's not totally out of it, yeah. Because when you do the recycling, we have maximum of recycling. Yeah, for information, for the EOPO that recycle only maximum up to 5 times. After 5 times, you need to top up with a new chemical, new EOPO, so that you can perform a better efficiency. Right? This is something that I would like to share with you guys.
So question for maybe poster, you can also see able to see that.
Yeah, microalgae can be used as a nutrient deficiency of human or animal, which is better. Right, I can say both also better, depends how you cultivate the microalgae. Right, because I'm in Malaysia, we use this microalgae used as animal feed as well as human supplement. So depend how you cultivate. Of course, if you use animal feed, you're not required to have very clean environment. While if you use for human consumption, you need to have very control and very clean environment. You need to follow a lot of protocol, a lot of procedure SOP. So this is the different things. Yeah, depends. Yeah, if you want to go for higher end, higher market, definitely you go for human being consumption, which is food supplement, vitamin, medicine. While if you use for animal feed, no harm, you can test it out. Yeah. So depend what is the end product that you want. I can say both also possible. Right. I hope I answer your question. Yeah, welcome.
Yeah. Okay. So any other question from your side?
I have no question.
Yeah. I think the people will contact you for publication because you are recently hosting out of journal. So
Don't
the
worry.
people are.
Yeah, I'm happy for any collaborations. Yeah, because in my opinion, yeah, research is need to be have a good collaborations. Yeah, no hero in this world. Yeah, everyone work together to get to provide a better world to live. Yeah.
Yes. Okay. Maybe all have done with their questions. Then we can proceeding for the next event. So please stay back, Doctor. So because we're gonna felicitate the event also. Yeah, we stay back and then I share. You can if you stop sharing, then I can. Yeah, you already stopped sharing. Sorry.
Yeah. You stop sharing. Yeah. So
Yes.
should I stay? Should I stay in this? I can leave. By the way.
No, you can stay back for 5 minutes.
Okay, no problem. I will stay.
Yeah. Okay. Formally, I'd like to thank Dr. Laksho for his excellent presentation, and then he very clearly answered all the questions raised by the participants. So with that, I'd like to thank him with the certificate of appreciation for his excellent talk delivered today. Thank you, Sho, for interest showing your presentations.
Yeah, welcome.
Yes. Okay. Now we are actually getting into this welcome to the inauguration for the Nano and Biomaterial Association. Actually it was emerged in a closed association, but with that interest of the participant, it is open now, and it's also pre-registration. The student and scientist research scholar can do contribute and then can be a member of this association. So where we can exchange the knowledge of nano and biomaterial stuff in this association. So that's what we're also working many on nanomaterials with other microbes and for refinery other stuff. So you're also the right person to felicitate and then inaugurate these things. So with that, actually, our one person. Okay. Yes. So formally, I'd like to welcome our president of this association and also the convener, our Professor Dr. S. Hemalada, to welcome and give this presidential note to this group. Yes, ma'am.
Good afternoon to everyone. I'm really happy to be associated with this Biomaterial Association. From School of Life Sciences, DS Abdur Rahman, Crescent Institute of Science and Technology. We are inaugurating this Nano and Biomaterials Association, which is NBA. This association will support the development of nanotech innovations that improve the lives of consumers, preserve our environment, and advance our world. We expect the members of MBA who can be committed to the safe, sustainable, and beneficial use of nanotechnology and nanomaterials across all industries. We work towards a better understanding of nanotechnology's important role in society and economic development and building a positive global environment for nanotech innovation. The mission of NBA is to support nanotechnology research by communicating the essential role of nanotechnology in improving our daily lives and advancing our nation and globe. Forming focused research collaborations with the stakeholders across the public and private sectors to help build a positive regulatory policy and acceptance environment for nanotechnology, identifying unique areas of research that can provide an unique and beneficial value, creating partnership opportunities between members that can help bring new nanotech innovations to the market. This is also to demonstrate our responsible commitment to developing and stimulating safe, sustainable, and beneficial nanotech innovations, helping our MBA members, especially startups, to deliver new products to market through Crescent Innovation and Incubation Council. The main objective of the NBA is to advocate for and organize the exchange of ideas involving the unique science and technology present in biomaterials at the nanoscale. By focusing on science, the NBA will champion the continual push to uncover new knowledge at the nanoscale and connect this to macroscale properties and behaviors of biomaterials. Through its focus on technology, NBA will foster innovative design and synthesis of nanobiomaterials useful in the creation of new and better devices, diagnostics, and therapeutics for biomedical applications, as well as the products related to agriculture and environment. The NBA emphasizes on an interdisciplinary vision to facilitate the translation of nanomaterials to achieve intended biological significance and medical impact. The vision is to establish NBA to become a thought leader in the nano research community by emphasizing nanoscale and nanoscience discovery, nanotechnology applications, and clinical translation innovation. From School of Life Sciences, we have published more than around 50 research papers in the area of nanotechnology and materials and its applications in medicine, agriculture, and as well as environment. And our students and faculty have filed 20 patents, and also all these 20 patents are published in this nanotechnology and biomaterials area. And our students, research scholars, and faculty have developed around 36 products in this area. Our undergraduate and postgraduate student research scholars, and faculty have incubated 24 startup companies in Crescent Innovation and Incubation Council. And out of 24, 19 are in the area of nanotechnology and materials. On behalf of School of Life Sciences and MBA, I invite all the participants and also your friends and collaborators to become a member of MBA. And this is to disseminate the information on nano and biomaterials applications among the society so that we can serve to the society in terms of applying the nanotechnology in agricultural environment and also to improve the quality of human life. We'll be circulating more information on various events that we'll be organizing through this MBA, opportunities that are available, and research opportunities as well as the job opportunities and collaborative opportunities. And whatever the courses that we are going to plan and conduct, maybe 1 credit courses, certificate courses also we are planning to organize. And we'll be circulating all this information to the MBA members. So please stay tuned and try to be a member of MBA. Thank you.
Thank you very much, ma'am, for your note, which contains all the detailed information on the association. Okay. Next is the question of inaugurating the thing. Actually, we are inaugurated the logo of NBA. So this is a logo, Nano and the Biomaterial Association logo, which can actually this different colors which indicate different nanomaterials. Actually, it's experimental results of quantum dots and then clusters and nanomaterials in the directions. So actually the object introduction of the thing is already Professor explained, but the point of view to explain the Nano and Biomaterial Association is an India emerged association with the potential visionaries in nano and biomaterial related research. So objective of is to familiarize the recent advancement and synthesis of nanomaterials, clusters, quantum dots, and biopolymers as well. And also to create the awareness on the importance of nanobiomaterials, to perform the high throughput analysis of nano and biomaterials and apply in different domains, to augment various outreach activities related to nano and biomaterials. So these are the office bearers of the association, the president, Dr. S. Hemalata here, and the secretary is myself, and the coordinator, Ms. Ranjani, scientific assistant. So who are the office bearers, and then the members can join through this link, and then the QR codes are provided. So for this, some of the known scientists would like to give their felicitation to this NBA. So I'd like to welcome Dr. Tajin sir for giving the felicitation address. We're sorry for the technical issue. Then we proceed with next felicitation by Professor Jungwon Kim from the International University. Actually, she could not join the meeting, but she sent her wishes to this association that I just waiting her heartfelt congratulations to the launch of Nano Biomaterials Association and best wishes for big achievement to come and collaboration between NBA and INU, that is International University, by Professor Jungwon Kim, Division of Bioengineering. So I'd like to welcome Professor Parlok Shaw to felicitate with a few words. You can unmute and talk.
Yes.
Yeah.
Is anything you want me to talk?
Yeah, yes. You are.
All right.
So yeah, you can talk to felicitate the event.
Sorry, what you want me to talk about? Yeah.
Yeah, just
a casual talk, is it?
Yes.
Okay. Actually, I'm not prepared
The
for
greetings.
anything. Yeah. But anyhow, yeah, just to thanks, yeah, for this organizing this interesting talk. Yeah. And I'm also happy to be part of this. Yeah. I believe there's a lot for me to learn from you guys in this Nano and Biomaterial Associations. So NBA. So feel free to contact us. Yeah, we have more collaboration. Actually, Malaysia, we have uncovered the chairing of this International Bioprocess Society. I believe it's highly related to this Nano and Biomaterial Association. So in future, we can have more activity together. Yeah, like joy conference, joy workshop, joy guest lectures, and so joy publication and exchange. Yeah, whenever there's possible. Right. I think there's a lot of opportunity and a lot of these things that we can do together. So I'm looking forward to that. Thank you so much.
Yes, thank you so much. Actually, I thought of asking you these things, but you are coming forward and then mentioning all jointly organizing events. And also, we welcome some advisory board for this our association where you can help us to lead this show, and then we have some special issue in your journals as well. Okay, thank you for giving the felicitation. Actually, Dr. Saravanan is there because he to join. I think his internet is not stable, so he will not. We will give it later. Okay. So we come to the end of this today. So I'd like to thank each and every one of you who participated this event for last 2 days. Right from the Rahman Sharif from the EMBL and then the European Molecular Biology Laboratory, UK, and followed by Dr. Saravanan Kumar Kandasamy from Kangwon National University, Korea. And today, Professor Parlok Shaw from the University of Nottingham for this lecture series. And also we inaugurated the association called NBA, where most of the members, showing the interest to participate as a member in that. And last year also the feedback was very good, and hope this today also it is an wonderful and enjoyable lectures given by the Professor Show. So with that, I'd like to thank. Actually, this is a kind of a summary of deliberations. Actually, totally we received 1,136 registration for that, and we are very sorry for that. We cannot accommodate everyone in the Zoom link. And then we made an alternative into the direction of our dean to go ahead with the live streaming. So that was a very good effort that all the participants can able to see through the YouTube link. And then there are various sectors. Mostly the students have came forward to register in a large quantity. Uh, and so this is YouTube link. In last day it was 953 views were viewed last day event. So I hope today's maybe some more views maybe could be there. So the formally, I like to thank our management for giving this up, giving us the support, and the Vice Chancellor, Registrar, our IAR officials, and the Dean SLS for giving the constant support to organizing these events and also inaugurating the association. And President and Secretary of Biotech Research Society India, and especially Dr. Binod, who is constantly giving support the workshop schedule to this all the participants. That's why we could got a huge participation in this. And also we thank Director CIC for providing this Zoom link platform, and especially Alicia, he made a very good effort, and Dr. Simon for his support of making thing happen, and all the speakers from various countries, because without them it's not possible, and along with the technical issue, because all they giving talk from their country, and we also understand our technical internet issues, and then the abroad. So with that, we successfully completed that. And lastly, and most importantly, the participant. So actually, the WhatsApp group, and then mail, and then Zoom, everything is overwhelmed. So that much effort taken by the participant to joining this event. So thank you so much for that, your support. We do organize many events, so we will circulate this information in the group. If you are be there in the WhatsApp, you can get this information further. So with that, this is end of this event, and you can find this feedback link here, and then I will post it in the group, WhatsApp group, YouTube, and also in this Zoom link. You can note it, and then you can you can submit this form for your certificate. Thank you so much, Dr. Show, for your presence up to now.
Right, no problem. Yeah.
Thank you. We'll meet soon.
Yes. Hopefully one day you can come to Malaysia. Yeah, I will host you here.
Yes, sure.
Okay, thank you everyone.
Thank you. Bye-bye. Bye.
Bye-bye.
I share the YouTube, feedback link now. Please wait a few seconds.